Okay. Assalamualaikum warahmatullahi wabarakatuh. Good morning, good afternoon for all. Her Excellency, the Vice Rector for Organization Szczecin University, Professor Kinga Blagagiozska, His Excellency, the Vice Rector for Planning and Collaboration Affairs, Universitas Brawijaya, Professor Muhammad Sasmito Jati, Honorable, the Vice Director for Science of the Institute of Marine and Environmental Sciences, Dr. Joanna Dudzinska Noak, Honorable, the Dean of Faculty of Fisheries and Marine Science, Universitas Brawijaya, Professor Happy Nursyam, Honorable, the invited speaker, Professor Andrzej Witkowski from Szczecin University, Honorable, the invited speaker, Ms. Alexandra Golubeva from Szczecin University, Honorable, the invited speaker, Professor Yunianta from Universitas Brawijaya, Honorable, the invited speaker and also the head of the Committee of World-Class Professor Program, Professor Yenny Risjani from Universitas Brawijaya. Respectable all Zoom and YouTube participants of this webinar from around the countries, such as Poland, Turkey, Saudi Arabia, France, Germany, Japan, Algeria, Vietnam, China, and Indonesia. Firstly, let's thank to the Almighty Allah Subhanahu wa Ta'ala, who has given us many mercies and blessings so that we can attend this international webinar without any troubles and obstacles, even in this pandemic situation. Secondly, salawat and salam may always be given to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us from the darkness to the lightness, from the stupidity to the cleverness. I'm Sulastri Arsad, your master ceremony and moderator of this webinar. So, ladies and gentlemen, we're delighted to extend our warm welcome, selamat datang, to all participants to the first international webinar, World-Class Professor Program, a collaboration between Universitas Brawijaya and Szczecin University, Poland. With the topic of the webinar today is Algae Biodiversity and Benefit. We would like to thank everyone here for taking time to attend our remarkable webinar. So, to start our opening ceremony today, allow me to read today's agenda. First, opening. Second, remarks. Third, photo session. Fourth, webinar presentation session. Fifth, discussion session. And the last, closing session. Those are the today's agenda, which will be presented soon. Dear audiences, ladies and gentlemen, let's open this agenda by reciting Basmalah. Bismillahirrahmanirrahim.
Bismillahirrahmanirrahim.
Excellencies, distinguished guests, fellow students, ladies and gentlemen, we are now going to invite Professor Yenny Risjani, Head of Committee for World-Class Professor Program, deliver the report of the program, as well as explaining about algae, a promising sustainable resources from marine ecosystem. Prof. Yenny, time is yours, please.
Thank you very much, Mbak Ella. Bismillahirrahmanirrahim. The Honorable, the Vice Rector for Organization, University of Szczecin, Professor Kinga Blagagiozska. The Honorable, the Vice Rector for Planning and Collaboration Affairs, University Brawijaya, Professor Dr. Engineer Muhammad Sasmito Jati, M.S. The Honorable, the Vice Director for Science of the Institute of Marine and Environmental Sciences, Dr. Joanna Dudzinska Noak. The Honorable, the Dean of the Faculty of Fisheries and Marine Sciences, Brawijaya University, Professor Dr. Engineer Happy Nursyam, MS. Good morning and welcome for all participants from Poland, from France, and from Europe in general, and also from Germany, and especially the inviting world-class professor, our colleague, Professor Andrzej Witkowski from the University of Szczecin, and also Professor Haas from Germany. And good afternoon, selamat siang for all Indonesian participants. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, thank God. We can be here together today, and we are very pleased to welcome you for the WCP program. This program is funded by the Directorate General of Higher Education, or DICTI, the Ministry of Education Nasional Republik of Indonesia. The objective of the program is to invite the world-class professor from the other countries to Indonesia to increase the quality of Indonesian researcher. That is the objective of the program. Last year, the WCP program included a mobility program, but for this year, because of the COVID-19 pandemic, the agenda has been changed. This year, the WCP program is scheduled from last August to last November, and it consists of 12 activities, amongst other, focus group discussion or FGD, workshop, and webinars. Depart from the Ghana project coordinated by Professor Jean-Luc Mouget from Nantes University, France, and after the second of Professor Andrzej Witkowski from Szczecin University in Brawijaya University, we continue to apply for the WCP program, and that because of the Ghana we can meet together here, and the theme of the program is Algae Biodiversity and Benefit. So why we have raised this theme, and for that reason, allow me to give an overview of algae in a general form, globally and rapidly. Please, for the presentation, okay, I will share. Share screen. Yeah. It is okay. Can you see?
Yes, everything is okay.
Yes. Okay. I will slideshow. Slideshow. I will make. Sorry. Okay. We'll start. Algae. This is my my presentation about algae, promising sustainable sources from marine ecosystem. What are algae? Algae are simple plants that can range from the microscopic or microalgae to large seaweed, macroalgae, such as giant kelp, for more than 10 meters in length. Algae can be grown using water resources such as brackish, sea and wastewater unsuitable for cultivating agricultural crops. And algae are very diverse and found almost everywhere on the planet. They play an important role in many ecosystems. The benefit, algae are believed to be a good source of renewable energy. I will give you 10 reasons why. And algae are emerging to be one of the most promising long-term sustainable sources of biomass and oil for fuels, food, feed, and other co-products. So nearly all these benefits stem from the fact that these plants have evolved over billions of years to produce and store energy and other valuable natural products. They do this more efficiently than any other non-natural of engineered process. This is the reason why algae are promising new source of renewable energy and other products. First, algae grow fast. The algae grows quickly. It's about 30 to 60 times faster than land-based plant. Microalgae can double their number every few hours, can be harvested daily, and have the potential to produce a volume of biomass and biofuel many times greater than that of our most productive crops. Second, algae can have high biofuel yield. Algae store from oil, yes, carbohydrates combined with productivity, then can produce to as many as much of billion gallon per acre per year. From this process, for example, algae and site selection, algae cultivation, harvesting, biomass processing, oil extraction, and then biodiesel production. I will make. Okay. And algae consume carbon dioxide. Like any other plant, algae consume or absorb carbon dioxide as they grow, releasing oxygen that is needed for living organisms. This is the global temperature of our world from 1880 to 2090. The temperature and the carbon dioxide rise. Yeah. It could become a weapon in the fight against climate change. Natural algal population may explain how environmental change affect global carbon cycle. When in balance, the total carbon dioxide removal, this is the cycle of carbon dioxide, and human activity since industrial revolution has increased drastically carbon dioxide concentration in the atmosphere. And microalgae in the ocean absorb 1/2 of carbon dioxide in the atmosphere produced by human activity. Due to carbon dioxide absorption, algae can be used as biolamp. This is a feature of energy. This is the French chemist is developing street lights that can absorb carbon dioxide 200 times more efficiently than tree. And this is an example of spirulina algae that can be used also for biolamp. The proposal of algae for street lamp has been designed by Peter Hovat from Hungary. He thought up a way to actually capture smoke with street lights that will turn into biofuel. This algae street lamp is eco-friendly and many levels as it lights up the streets with energy efficient LED bulbs, as well as it air pollution to make a cleaner burning biofuel before it breathes out oxygen. The algae street lamp will serve a double purpose by promoting cleaner vehicle and cleaning up after the fuel exhaust saturating in the city. 4. Algae do not compete with agriculture. Algae cultivation used both sealand that in many cases is unsuitable for traditional agriculture, as well as water sources that are not usable for other crops, brackish and wastewater. 5. Algae have valuable contents. Microalgae can be cultivated to have a high protein and oil content. For example, which can be used to produce other biofuel or animal feed or both. Microalgae biomass, which is rich in micronutrients, is already used for dietary supplement to advance human health. Algae have also valuable content used for feed, food, or as energy sources. This is product from algae. For example, algae oil. It can be for biofuel or skin or personal care products. From protein, we can get animal. We can produce for animal feed and for carbohydrate, for electricity generation and ethanol and other biofuel. And from biomass, we can get solid fuel and aquaculture feed. 6. Microalgae can be grown in the sea, pond, or in wastewater. This is macroalgae grown in the sea or in the pond and in wastewater treatment. We use it as a bioremediation. 7. Algae can purify wastewater. This is algae thrive nutrient-rich waters like municipal waters or sewage, animal waste, and some industrial effluent. At the same time, purifying this water while producing a biomass suitable for biofuel production. 8. Algal biomass can be used as an energy source. This is algae with help of sunlight and with copper and zinc that can be become a biobattery. For example, if one biobattery with other biobattery, we can get more energy, and this means sustainability. After oil extraction, the remaining algae biomass can be dried and pelletized and used as fuel that is burned in industrial boiler and other power generation sources. 9. Algae can be used to produce many useful products. Algae can be cultivated to produce a variety of products for large to small market. For example, biofuel, for example, biodiesel, biobutanol, bioethanol, biogas, or bioplastic, feedstock, animal feed, fertilizer or nutrients, food, and pharmaceutical or vitamins. And also for cosmetic. For example, for plastic, it can be for plastic bottle, plastic cup, food packet, food container, plastic dishes, sachet. Yes, algae are also being considered as a feedstock for the production of range of sustainable chemicals from plastic to fertilizer. For fertilizer, seaweed have been used as a fertilizer for centuries, and some microalgae fix atmospheric nitrogen and could be a source of organic fertilizer, or we call this green manure. The production of industrial chemical and fertilizer from microalgae is still in its infancy, with many more breakthroughs still to come. Cosmetic. The natural cosmetic for 4 years has been leveraging algae in products focused on skincare. Companies are now developing this compound into products that offer humans the same benefits. This will be a fascinating industry to watch in the coming years. You can see several products from sun chlorella and crème de la mer from France. And for example, this is the product Algenis from USA. And also this is the famous one from France. Yeah, I use this. Okay. Again, for human nutrition and health products. Food products that are currently produced from algae include food ingredients such as essential omega-3 fatty acids used in infant formula, carbohydrates, and emulsifiers produced from seaweeds and food supplements. Spirulina, produced in a large pond in California and Hawaii, is a traditional food source in several places around the world, including in Mexico and Africa. We can see, for example, climate, blue-green algae, chlorella. This is from chlorella, spirulina, and omega-3 for health, immune system, and brain health. Yes, and from brown seaweed also. And for specialty and bulk animal feed, because they contain carbohydrate, protein, and vegetable oil, in particular omega-3 fatty acids, microalgae have great potential to produce sustainable animal feeds. They also contain important micronutrients and vitamins, as well as valuable pigments used in animal feeds, such as beta-carotene, lutein, and astaxanthin. As a result, algae have emerged as exciting sources for higher value specialty animal feed. Algae have been used as a source of feed in aquaculture operation for decades, including as feed for fish and shellfish like clams, oyster, mussel, and scallops. An estimated around 30% of current algae production is sold for animal feed application, with poultry and fish are the main targets. And the algae industry is a job creation engine. Algae can grow in a wide variety of climates in a multitude of production methods, from ponds to photobioreactor to fermenters, and thus will create a wide variety of jobs throughout the world, from research to engineering, from construction to farming, from marketing to financial services. In the USA, the Algae Biomass Organization projects the potential for creation of more than 200,000 jobs in this sector by 2020. In Australia, for example, revenue from processing the seaweed could add a further about $250 million per year to the economy and support 1,200 South Australian jobs. In Indonesia, tourism sector has been slumped because of COVID-19, and they converted their job profession as seaweed farmer. This is an example. I found the news from Indonesian newspaper. Pelaku wisata Bali beralih profesi jadi petani rumput laut. That means there are many workers who works in the tourism sector before the pandemic, as for example, as tourism guide or driver, and at present they become seaweed farmers in Bali, for example. And how about the relation with COVID-19 pandemic? We found the review about algae related to COVID-19, novel coronavirus pandemic in 2020. Seaweed to ratio. Why does substantial supporting research about the antiviral property of seaweed polysaccharide seems to be unrecognized by the pharmaceutical community in these disparate times? That's the reality. But, we know that many species of marine algae contain significant quantity of complex structural sulfated polysaccharides that have been shown to inhibit the replication of enveloped viruses. There are many compounds. For example, Vicoloid, from Vicoloid, from polysaccharide, or other sulfated polysaccharide extracted from green algae or from brown algae. We found also from Canadian researcher, they use algae to produce COVID-19 test kit. And also from university, Western University researcher, they produce, they use algae to help quickly produce COVID-19 test kit. How about Indonesia? In Indonesia, we found antibacterial hand sanitizer from seaweed extract combination that originated from Equima spinosum or Equima cottoni. Yeah, the conclusion. Algae can contain high level of oil, carbohydrates, sugar, and proteins, and can be used to produce renewable fuel, animal feed, and even human food. In fact, algae, both micro and macroalgae, or seaweed, are already widely used in food products, providing texture, stabilizing features, and important nutrient for health. In short, fuel, feed, and food derived from algae can help provide the answer to many of our pressing problems. Related to COVID-19 pandemic, it is also a promising source. That's why billions of dollars are being invested in the research and development of algae-based technology. It won't be long before fuel made from algae will start replacing petroleum fuel in our tank. Indeed, petroleum in large part derived from algae that grew millions of years ago. We now need to replace this old algae with new algae fuel. Thank you very much, and enjoy the event. Thank you.
Okay, thank you, Prof. Yenny, for the explanation of the VCP program and also the introduction of microalgae. Moving right along, we are now going to listen the opening remark delivered by the Vice Rector for Organization, Szczecin University, Professor Kinga Plaga Georgiska. Professor, please, time is yours.
Thank you. Dear Rector, dear Dean, dear ladies and gentlemen, it was a great pleasure to accept the invitation to open your scientific webinar. The collaboration between Universitas Brawijaya and the University of Szczecin is an example of collaboration which breaks geographical barriers. The University of Szczecin is a border university that grows on the traditions of multiple cultures that moved through this region as a result of the tragedy of the Second World War. It makes us open for other nations, including the international cooperation for the welfare of science. We want to know the history and the culture of different nations who are geographically distant, however so close in terms of peace-loving attitude. The great Indonesia is a pattern of an impressive history. We can draw multiple timeless values from it. The pandemic has shown us that we are even closer to each other. We can still meet and work together. I hope that your cooperation will be a source of inspiration for other scientific from our universities. We can learn how one can develop friendships and at the same time deepen research achievements. I think it concerns above all deepening our bonds with the Universitas Brawijaya. We want to develop the cooperation with you in various fields, This beautiful motto, building up noble future, is also very familiar to us. Striving for perfection is a challenge in which we can support each other. Today's webinar is an event extremely important for the future of our countries. It touches terms of the fundamental impact on our planet. As a lawyer, I may not know much on algae, but I know how important biodiversity is for saving our planet. This problem is especially close to us due to our geographical position. 85 million people live around the Baltic Sea, which makes it one of the most populated areas of this kind. Fishing and sea transport of the Baltic Sea are one of the most developed in the world, and they destroy its resources. Changes made by man greatly exceed the durability of the sea environment and endanger its small biodiversity. The Baltic Sea ecosystem is very sensitive to pollution and negative environment pressure. Our sea has been given to status of the particularly sensitive sea area. It shows how important is your joint research and recurrent scientific undertakings which come from the high practical necessity. It is great honor that I open this webinar, one of the very interesting cyclical meetings. I wish you all fruitful discussions, excellent results, and satisfaction that comes from your commitment. Thank you for your attention.
Thank you, Prof. Kinga, for the remarkable speech. Thank you. Next.
I'm sorry, but I must quit from this meeting because I have a lot of responsibilities connected with pandemic situation on the university.
Yeah, we understand, please. Thank you, Prof. Kinga.
Great discussion.
Thank you very much, Prof. Hinga.
Thank you.
Thank you very much.
So next, I'm honored to invite the Vice Rector for Planning and Collaboration Affairs, Universitas Brawijaya, Professor Muhammad Sasmito Jati, to deliver an officiating speech. Please.
Assalamualaikum warahmatullahi wabarakatuh. Our honorable guest, Professor Kinga Flaga Guiresinska. Sorry, my mistaken spelling. Vice Rector for Organization, University of Szczecin, Poland. Professor Joanna Dudzinska-Nowak, Vice Rector for Science of Institute of Marine and Environmental Sciences, and my colleagues, Professor Happy Nursam, the Dean of Faculty of Fishery and Marine Science, Brawijaya University, Professor Andrzej Witkowski, Professor Yunianta, Professor Yenny Risjani, Alexandra Golubeva, respective researcher, respected senior scientists from Japan, France, and Saudi, dear colleagues, lecturers, ladies and gentlemen, as well as my beloved student. Hopefully Allah give you mercy and compassionate and healthy condition. Amin. The first international collaborative webinar between Brawijaya University Indonesia and Szczecin University Poland, entitled Algae Biodiversity and Benefit. This webinar will be synergizing knowledge between two universities with more than 10,000 mile distance that we cannot imagine it before. Two universities with such distance could be gathering closely. We have gathered here to discuss with enormous topics such as algae, biodiversity, and the benefits. I am highly honored to take this opportunity on behalf of Brawijaya University to say welcome to all of you to this seminar online. I just want to say good morning for Poland and France, and good afternoon for Indonesians and Japanese. We have believed this conference attending with several distinguished personalities who are highly qualified. We have shared and care discussed global problem that all of us concern, what we call algae. If we think about conservation, that's mean worldwide and humankind responsibilities. All of us should be made attention about this topic, and we believe to conserve biodiversity itself, we cannot live in the benefits, even though logical way of thinking of beneficiation is something pragmatically, but it is used to be. Moreover, for Indonesian side, as the largest tropical archipelago countries in the world, that we are living with more than 70,000 of islands, I believe that one beneficiation to discuss. We have gathered here to discuss and solve the global problem, humankind problem, more after the pandemic of COVID-19. We just talk about very small unseen creature, but it is giving a great impact to our daily life, our habits, and more important thing is our way of thinking about what is life mean. I think this great moment to rethinking about the safety of humankind life. I hope you all will find this seminar highly engaging, fruitful, and beneficial for our future, our happiness and prosperity. And the last but not least, I just want to say thanks to organizing committee, Professor Yenny Risjani, that always keep the seminar going on, even in pandemic circumstances. This seminar should be held in any condition and circumstances. Hopefully all of you will be enjoying this meeting. May Allah bless you, Amin. Today, in the name of God, I officially open the meeting with saying Bismillahirrahmanirrahim. That's all, and thanks to all of you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for a meaningful speech and officiating this event. Furthermore, we are going to listen to the opening remark delivered by the Vice Director for Science of the Institute of Marine and Environmental Sciences. Please welcome Dr. Joanna Duzinska-Noak. Dr. Joanna, time is yours, please.
Thank you very much for invitation. Ladies and gentlemen, good afternoon in Indonesia and good morning in Poland. It is a privilege and a great honor to welcome you at the first webinar within World-Class Professor Program, which is for us a next step after a teacher exchange, next step of collaboration between Brawijaya University and Institute of Marine and Environmental Sciences, University of Szczecin. Such collaboration is very important, especially for the university's community, because in addition to the exchange of experience and knowledge, links science and teaching. This collaboration is like a bridge that connects and allows to spread knowledge around the world. So we are very happy to be a part of this program, of this webinar, and I wish you very interesting lectures, fruitful discussions, leading to many brilliant ideas of further collaboration between our universities, and which will result in joint future scientific publication. Please enjoy the meeting. Thank you.
Thank you.
Thank
you very much.
Dr. Joanna. Thank you. So now we are going to listen. Thank you, Dr. Joanna, for the great opening speech. And now we are going to listen to welcome speech delivered by the Dean of the Faculty of Fisheries and Marine Science, Universitas Brawijaya, Professor Happy Nursyam. He will present about the profile of Faculty Fisheries and Marine Science, Brawijaya. Please, for Prof. Happy, time is yours.
Okay. Thank you, Ibu Sulastri. Good morning for Poland and friends, and also good afternoon for Indonesia. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Rector of Szczecin University, Poland, and the Honorable Vice Rector, Dr. Joanna, the Honorable Vice Rector of Collaboration and Planning under University of Brawijaya, and also Professor Yenny Risjani as Head of Bioecotox Research Group under Faculty of Marine and Faculty of Fisheries and Marine Science. All of speakers, Professor Witkowski, Professor Yenny Risjani again, Professor Yunianta, and also Dr. Alexandra as a speaker will be present today. Distinguished participants, ladies and gentlemen, first of all, I will let us pray in the presence of Almighty God for His blessing we can gather on this day. It is event of webinar activity by BCP program, world class professor program, University of Brawijaya, especially on algae, biodiversity, and benefit. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Happy.
Yeah.
Thank you, Professor, for such interesting
Yes,
information
thank
about
you.
the Faculty of Fisheries and Marine Science. And thank you also for your time to attend this meeting. Ladies and gentlemen, all participants.
How to close this?
You can stop share screen.
Okay. Yes, thank you.
Okay, thank you, Professor.
Yeah.
So, ladies and gentlemen, as we have finished our opening remarks, we are now continuing our meeting to the next agenda. However, we could take photo together by screenshot the screen. So please, to all participants, to open your video and camera, and for the committee, Theo, please help me to take photos by screenshot. Theo?
Okay.
Okay. So we can start?
Yeah.
You
Open
can
the
count.
video, please. Open the
You can
video.
count, Theo.
Okay.
Okay, we can start.
Let's take a picture. I will
Yes.
count from 3. Smile. 3, 2, 1.
Okay, next slide.
Slide.
We have how many slides? Oh,
Yeah. Smile.
Cool.
Okay, done. Thanks.
okay. Already finished?
The last
Yeah.
photos must be
Thank you,
cry.
Theo. Yeah, Professor?
No. Just.
Okay, thank you. Dear audiences, we come to the main agenda, that is webinar. The keynote speeches which will be delivered by, first, we are honored to introduce our first keynote speaker, Professor Andrzej Witkowski from Szczecin University. And here is his brief profile, please. Okay, so we can read in this slide. So Prof. Andrzej is a lecturer and a professor and also a scientist at the Institute of Marine and Environmental Sciences at the University. And he has graduated his PhD at the University of Warsaw, and now he works in Szczecin. And he has an expertise on paleo aspect of marine diatoms and species, biodiversity, taxonomy, phylogeny, biogeography of marine littoral diatoms. And now his current research about molecular phylogeny and diatom research. Okay, and for today, Prof. Andrzej will deliver the presentation entitled Exploring the Biodiversity, Biogeography, and Taxonomy of Indonesian Archipelago Diatom Assemblage from east to west, from north to south. So please, Prof. Andrzej, time is here for 20 to 30 minutes ahead.
Unmute. Good afternoon, Mrs. Chairman.
Good afternoon.
Good afternoon, ladies and gentlemen, dear colleagues, friends from Indonesia and worldwide. I think we also have South Africa. Tommy Bornman, at least I remember he was ready to join us. So please note, I added a remark, raw data, because we have just finished analyzing the data, and what is maybe shocking, I shall say, we shall acknowledge the virus, the coronavirus, because I was sitting at home and I was getting the diatoms from Indonesia. And one I shall stress, it would be probably not possible, our cooperation, without effort of our colleague, our friend, Jean-Luc Mouget, Professor Jean-Luc Mouget, who who has organized a team of international scientists for the European Commission funded project on marine blue diatoms from the genus of Haslea. And from this not very distinct photo, you can see what kind of habitats we are working on. And many of us have indeed dreamed.
I think we don't see your presentation.
Oh, you don't see?
No.
How? Okay. Who will then guide me how, okay. Is this, and now?
Yes.
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 It's working, okay.
Yes, it is.
Sorry. So from this image, you can see what kind of paradise-like habitats we have been sampling and studying. Why Indonesia? Of course, not only because of this spectacular views, but and not only because of spectacular habitats. Indonesia, as you can see, is surrounded, it was mentioned, it's thousands of islands, and the coastal waters of Indonesia are very productive, are very rich in terms of biology. why not studying diatoms there? If diatoms are, we know that microalgae which are sustaining or dominant in terms of primary production in the coastal waters surrounding Indonesia. This is one point. The other point was that some time ago we have been lucky to study diatoms from some of the outstanding places in the world in terms of biodiversity, which are named biodiversity hotspots. So our first experience was Madagascar, where we found it's really very rich. And Madagascar will be some kind of background or reference for what we have done in Indonesia. But for us, it was tempting to see how the diatoms perform in Indonesia, especially that, as you can see, in the worldwide recognized biodiversity hotspots from Indonesia, we have two biodiversity hotspots: Sundaland, which is the western Indonesia, and Wallacea, which is the eastern Indonesia. So this was tempting to have a look at diatoms from these sites and to test whether the diatoms are also showing this separation of the two biodiversity hotspots. Please note is next one just north of. So the whole area is really like a treasure, not only in terms of beautiful landscapes. Yes, so sampling have been done in 2014, in 2018, and in 2020, just finished this year. So we have sampled first South Java, the spectacular place named Tigavarna, which is the south coast of Java. Then I should say I was happy to see Komodo, and it was maybe an opportunity to fight with the dragon, but we let him, we let the dragon live in peace. So this is now not possible for tourists to go there, maybe possible to sample. So this is the National Park Komodo, is Pink Beach Island. So this is island of Flores, and this is Rincha, and this is Tigavarna. Then we have sampling in 2020, which we expanded to Western Kalimantan, West Kalimantan, which is Borneo, the islands of Lemukutan, and we expanded the sampling to more, let's say, anthropogenically impacted waters of North Java, of the area of Probolinggo, where the University of Brawijaya has the field station. And in 2014, there was some islands in southeast of Sulawesi, which have been sampled by our colleague Zhenya Gusev from Russia.
Next, let's welcome our second presenter, Professor Yunianta. He will present his speech entitled "The Potency of Brown Algae as Marine Rocks Raw Material." And Andrzej, could you stop the screen? The share screen?
Yes.
The share screen? Okay, thank you. So here the brief profile of Professor Yunianta. Professor Yunianta is a lecturer and scientist at the Faculty of Agriculture, Agricultural Product Technology, Universitas Brawijaya. And if you are interested to contact with him, his email is Yunianta@bsid. And he was graduated from France for his master degree and doctorate degree. And he has an expertise on using macro and microalgae for food, functional food, and pharmaceutical purposes. And for current research, he is active to make a research about bioactive from macroalgae and microalgae. So, for Professor Yunianta, time is yours for 20 minutes ahead, please. Would you share your presentation by your own, or please unmute your Professor Yunianta. Please unmute first.
Okay. Okay. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning and good afternoon. Good morning to our colleagues in Poland, France, and others, and good afternoon to our colleagues in Indonesia. This morning, this afternoon, I would like to present you about the result of our research, especially about the potency of brown algae as marine drugs raw material. This is the, you know well about this category. They are macro and microalgae, and I don't want to enter too much to the this category, but I only want to say that this afternoon I would like to share you about the result of our research about macroalgae. Maybe in the next webinar, I would like to talk about microalgae. In my research, I use brown algae. We have several types of brown algae in Indonesia, and I think brown algae sargassum is only one of type that is very little in comparing with other brown algae. That's very much in diversity. Yeah. I use this brown algae, Sargassum ristifolium, to investigate the bioactivity, especially about the anti-cancer bioactive. We know that in the brown algae there are polysaccharide, alginate, laminaran, coitan, etc. And also we have small compound that is soluble in organic solvent like methanol and hexane, etc. And all of these compound, polysaccharide and small compound in brown algae is, I think we only do very small research about that, and we have still have many opportunities to develop our research about this brown algae. This is the outline of my research. I extract and with methanol extraction, and then we fractionate it in organic solvent like N-hexane, acetic acetate, and butanol to get the different fraction. So we get N-hexane extract, ethyl acetate extract, and butanol extract.
Now, ladies and gentlemen, let's welcome our third presenter, which will be the last presenter on this webinar. So please, Ms. Alexandra Golubeva, present the material entitled Valuable Product of Cultured Microalgae. And here is her brief CV about Ms. Alexandra. So she is now a Ph.D. student at the University of Szczecin in the second year or the last year, Ms. Alexandra?
Second year.
In the second year. And her research interests about microalgae, of course, and the application of microalgae. And currently topic of the research about optimization of growth condition for scaling up the process of culture-based use of diatoms for valuable chemical compound and 3D structure biosilica. So please, Ms. Alexandra, time is yours for 20 minutes ahead. You will share screen by your own? Okay, okay, thank you. Perfect.
Good morning and good afternoon, dear colleagues. My name is Alexandra, and I'm a PhD student in Szczecin University. And now I'm going to show you some information about different substances from microalgae. So what is the process of culturing these microalgae and how we can get these valuable compounds? So we can grow microalgae under different stress conditions. This can be nutrient starvation, salt stress, temperature stress, light stress. After that, we can harvest it and fracture it. For example, centrifuge the biomass and take only biomass, not medium. And after that, we can fraction it on, for example, lipids, carbohydrates, proteins, pigments, and other. And after that, they can be used in different fields of biotechnology. So I will start with microalgae as a source of lipids. Microalgae can produce many different classes of lipids. They can be grouped in two main categories, structural lipids and with high content of polyunsaturated fatty acids, and storage lipids, mainly the form of triacylglycerols, made of saturated fatty acids and some unsaturated fatty acids. Lipids and fatty acids content of microalgae from microalgae species specific and varied widely among different species. Lipid content of thousands of microalgae studies show huge variation in ranging from 1 to 70% of dry biomass. On this slide, you can see the different classes of microalgae which can accumulate high amount of oil. For example, Bacillerophyta with amphora with 70% of dry biomass. Chlorophyta with Dunaliella and Neochloris, and Triboxyophyta with Nanochloris and of course Chlorella. How these lipids can be used in technology. For example, in cosmetic industry, ethanolic lipid extract derived from microalgae are used for lipid-based cosmetics such as creams or lotions due to the provision of both nourishing and protecting effects of to the skin. Furthermore, microalgae have been suggested as a potential feedstock for production of bioenergy, such as biodiesel. Biodiesel from microalgae has been reviewed extensively by some authors. The potential of this algae as biodiesel feedstock is due to the capability to accumulate a high amount of oil, and they can be converted into biodiesel. The difficulty of this production is mostly associated with high cost of, with high operational, maintenance, harvesting, and conversion costs.
Okay, thank you, Ms. Golubeva, for your great and nice presentation about microalgae and its valuable product. And thank you very much for all presenters for their informative and interesting talk. Very well, ladies and gentlemen, now we come to the question and answer session. And before that, I would like to remind you, all participants, to fill the attendance by clicking the presence link in the chat room on the right side and fill the form in order you can get the certificate for this webinar. So there are several questions already in chat room and from YouTube, and I will pleasure to read or maybe to invite the questioners so they can ask directly to the speaker. So first, maybe I will invite Ibu Umi Zakia. You are still here, Bu Umi? Bu Umi Zakia?
Yeah, thank you.
Okay.
Thank you.
Yeah, Bu Umi is a colleague in our faculty from the same department. So please, Bu Umi, if you have a question to Prof. Andrzej. Prof. Andrzej, yeah. Please unmute first. Okay. Please, Bu Umi.
Thank you for, yes, thank you for the opportunity for me to ask questions, Ibu Ella. Prof. Andrzej, your research is very interesting. And because I also once gave a lecture on about plankton or about microalgae. It was very useful for me to get the information regarding your research. My question is, you know that Diatom is one of the microalgae which is responsible for HAPS, harmful algae bloom. And when it happens, this phenomenon, usually the cell will be sink and settled in the sediment. And my question is whether you would know how long is the period usually this cell is sink and then become or give an outbreak again later on. You know, as we know that the HAPS phenomenon is always like a repeated in certain period of time, and maybe you can give me informations. I think that's my question, Sibuela. Thank you so much.
Thank you.
I'm not sure I will be able to answer because we are not working with hubs, with the let's say Pseudonychia, which and Tunichia, which produce domoic acid, but I can imagine they are the mass development of the bloom of the Pseudonychia is released by the combination of factors, nutrients and temperature. So this will be in dry season where the temperature of seawater increase and the possible nutrients, the salinity, temperature, and the nutrients are increasing. And also there must be the upwelling effect, I can imagine upwelling, which is releasing the nutrients. They will survive as long as the nutrients excess is not depleted. So I would say few days, maybe 1 or 2 weeks. And the spread is very fast. We have observed this in South Africa, from Port Elizabeth to Cape Town. In 3 days, the red water effect was has spreaded, but it was not diatoms, it was dinoflagellates. So I can imagine that diatoms are very quickly spreading. Fortunately, in Indonesia, we have seen only a few Pseudonychia species, which are potentially domoic acid producers, but they never achieve high, even in plankton samples, Pseudonychia was sporadic. That means that it's still good water quality and good conditions in the marine waters, preventing from the mass development of toxic diatoms. Sorry, it is probably not what you have expected, but to summarize, in rather sudden appearance of the bloom and the duration, possibly a few days up to very few weeks.
Thank you. Thank you.
It was the combination of temperature and the nutrients.
Thank you very much.
Thank you.
Okay, thank you, Prof. Andrzej. Bumi, I hope you get the point. And there is a question more for Prof. Andrzej. It's from YouTube, from Abigail Moore. And this is really fascinating. May I ask, I will have an access to the presentation or the recording, and maybe this is all the participants want to access the file presentation. However, we don't share the file, but if you have or you interested more far about this topic, the participant could contact directly the speakers. Yes. So you can contact by their email. Okay, and then yes, Prof. Andrzej, from Abigail Moore.
We
has any of yes.
We are open to any contact, direct contact. In 2-3 months, it will be accessible because we are working on the publication. The results are unpublished, so we would not. But if any kind of expertise is necessary, I'm open. And my colleagues in the lab are open for any kind of cooperation.
Okay, thank you, Prof. Andrzej. And next, is there any question for Prof. Andrzej here? Maybe you can raise your hand from the colleagues from Germany or Turkey, if you are still there, or Poland, from the fellow students. Okay. On waiting, we will continue to Prof. Yunianta. There is a question from Ph.D, our colleague also from Unpad, Pajajaran Bandung. Ph.D, are you there? Pak Ph.D?
Yes.
Ph.D. Oh, must okay. Please, Ph.D.
Yes. Okay. Thank you very much for the opportunity. It is really interesting webinar, and I'm really happy to see Prof. Andrzej, Prof. Yenny Risjani, Prof. Yunianta in this webinar. And I have several questions to ask before it is addressed for Prof. Andrzej, but now I'm going to ask for Prof. Yunianta first regarding the bioactivity of sargassum. According to your experience, Prof, is there any difference between the bioactive compounds from sargassum and their bioactivity when you sample from different regions, for instance, from west of Indonesia to the east of Indonesia? Is there any difference in the bioactivity of this species? Thank you in advance.
Okay, thank you for your question. Unfortunately, I use only from Madura coastal, yeah. But in my opinion, the bioactive can be influenced by geography, because bioactive is a secondary product, metabolite. As a secondary metabolite, normally it is very influenced by the stress condition of the macroalgae. If there is a stress condition, it will produce some kind of metabolites. So it depends on the condition of environment. Yeah.
Okay. Thank you, Prof. Yunianta. Yeah. Okay. Is there possible to ask
yes,
more questions to Prof. Andrzej?
Please, please.
Okay. Thank you. Thank you, Bu Ella. You're very kind. Okay. Prof. Andrzej, how are you?
I'm fine. Thank you.
Yeah. Very good morning there in Poland. I have a questions regarding your presentation. It is really interesting, describing the diversity of the diatom from the west to the east of the world, and also the diversity of the diatoms in Indonesia. However, I have two main questions. The first one is, could you please elaborate more about the sampling method for the marine diatoms that is taken from the coral reef and also the sediments? Because so far I'm only working with the diatoms that are attached in the macroalgae like Padina, searching for the Haslea that we are working together. Yeah. And the second question would be for the morphological identification of these marine diatoms. According to your experience, what is the best washing method to keep the diatom still intact and clean for the better image acquisition? Yeah, thank you, Andrzej.
Thank you for the questions. Yes, we sample with a small test tubes, either Falcon or just simple test tubes. We take the sediment usually by the diver, or we take the sediment from the beach, like it was in Tigavarna, depending on the hydrologic conditions. If the sea is rough, we have to restrict to the beach. But if we have splendid conditions like it was in 2018 or this year in Sulawesi and Borneo, we just, the diver can, the scuba dive can take a sample from 20 meters and more. Our colleague Tommy Bornman provided us with living diatom samples from 60 meters in Mascarin Plateau, but it was dredged, I guess. Okay. So the tube And it's just a matter to take the sand and leave some free space for the air that they can breathe, the diatoms. Okay. Otherwise, they will die immediately. The other issue is scrape. So we use the toothbrush to brush the coral, brush the Holoturia atra, brush any object which is submerged. We use also plankton net.
Yeah.
20 micrometer, but also 10 micrometer. And in the lab, we keep a live sample and we separate part of sample for the cleaning process, which is, we use a gentle, good, friendly for the environment method. We use weak hydrochloric acid, which is removing the calcium carbonate, and we use hydrogen peroxide. If you boil the sample in hydrogen peroxide, you get finally water. So it's friendly for the environment, but you can also use very strong acids like concentrated sulfuric acid, nitric acid, like a mixture of acids. So it's many different ways, but as I said, I prefer an easy method for the human health, for the lab colleagues, and for the environment.
Yeah.
And we wash the samples with deionized water, always distilled water. Take care about the tap water because you can get freshwater diatoms in the marine sample.
So it will contaminate our samples. Okay, thank you so much, Andrzej, for your explanation. Yeah.
Pleasure.
Take care. Yeah.
Okay, thank you, Pak Vidi. I hope you get the point. And thank you, Prof. Andrzej. And next, the question for Ms. Golubeva. Here in chat room, Pak Dr. Pak Abe Susanto. Are you still here, Pak Abe?
Okay.
From Undip.
Yes. Yeah. Thank you, Bu.
Please, my professor, please.
Okay.
Abe.
My question is easy. Yeah. Special for Dr. Alexandra. Yeah. My question is about the what kind of the microalgae that is the freshwater or marine algae. This is the first. The second is you explain about the picocyanin, I think. How to get the picocyanin, because the picocyanin is very sensitive with the light or etc. And if I can get the information, more information about the methodology, could you please a little bit explain about that? Okay, thank you very much.
Thank you. So the first question about seawater or freshwater. In my presentation, I talked about the seawater, algae, and freshwater. For example, I work with the freshwater in Moscow, but now I work with microalgae from seawater, and I know it's a little different from. They have differences, of course, but I prepare the presentation about all of them. Doesn't matter the seawater or freshwater, cyanophyta or xerophyta. So I just take them all and speak about them. And the second question, as for me, I unfortunately didn't have the chance to, by myself, to have this experiment on fuckable indetermination. But I know that I think it's with after we crushed the crushed cells of macroalgae, we used the phosphate buffer, right?
Okay.
Yeah, we used a phosphate buffer and we frozen it, and after that just some we just repeat these steps, and after centrifugation we take the supernatant, and I think what about light sensitivity? I don't know, because I didn't have time to do it by myself, but I have this I can send you this method which I found, the theoretical method, so if you want.
Okay.
So yes.
Okay. Clear. Clear. I will contact you again, more detail. I will would like to discuss. Yeah.
Yeah.
By email. Okay. Thank you, Alexandra. Thank you, the committee.
Thank you.
Please.
Yeah. Thank you, Pak Abe, for the question, and thank you, Alexandra. So I think this is the last question because we are limited by time. Ladies and gentlemen, finally, finally we come to the end of this webinar. So we would like to say thanks again for all the presenters for their informative and interesting talk, and to all participants, audiences, for your active participation. Hopefully the presentation will be beneficial for everybody. And also I read this in chat room. There is a earthquake in Ambon right now, so we pray for them, in Maluku, Ambon. And as information, we will hold 3 more international webinars until November 2020. So please, if you want to join us for the next meeting, you can contact the committee. Then last, I'm Sulastri Arsad as the Master Ceremony and Moderator today. With all the world-class Professor Program Webinar Committee, beg your pardon of the mistake we have made. And let's close this agenda by reciting Hamdalah. Alhamdulillahirabbil alamin.